Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Dear audience, welcome to all of you. My lecture will be about biomedical data analysis and processing using artificial intelligence. First of all, I would like to express my sincere thanks and appreciation to the administration of the National University for Science and Technology for organizing this important and update symposium program about artificial intelligence in medicine. I especially thank Professor Muhammad Al-Shafi'i, Dean of the College of Medicine and Health Sciences, for the good organization and follow-up. I thank as well all organizers, moderators and speakers, and wish all the success for this event. My name is Abdul-Qader Abbady. I hold a PhD since the year of 2005 from University Louis Pasteur in France. My specialty is in life sciences and molecular immunogenetics. I am a research director at the Atomic Energy Commission of Syria. I am the head of the Division of Molecular Biomedicine. Our main project is about the genetic engineering of camel recombinant antibody fragments called nanobodies and their applications as molecular tools for treatment and diagnosis of many diseases spread in Syria. I am a professor at the International University for Science and Technology and professor at the Syrian Virtual University for the MSc Bioinformatics program. I am also an editor of the prestigious Frontiers in Immunology scientific journal, which is one of many important journals belonging to the Frontiers Publishing Group. For more details about my scientific profile, I invite you to visit my pages on Google Scholar and ORCID using these active URL links. You can use this QR code to directly access this lecture in order to benefit from the embedded URL links and references. Section 1. Preface to Artificial Intelligence. As a century of technological advances, the 21st century is characterized by breakthroughs in informatics that have holistically changed our perception of data analysis. Although in early days the primary challenge was gaining access to information, nowadays rapidly growing data sets are available anytime in the scientific community and can be easily analyzed, shared, and stored in databases. In recent years, the advent of new experimental methodologies for studying the high complexity of the human genome and proteome has led to the generation of an increasing amount of digital biological and medical information. The completion of the Human Genome Project in the early 2000s generated a tremendous amount of biological data that paved the way for the emergence of bioinformatics. An analysis of these vast stores of information has revealed significant aspects of human genes and raised expectations about the complex nature of genomes. Bioinformatics is an interdisciplinary field that combines computer science, biology, physics, chemistry, mathematics, and statistics. Is now playing an essential role in the analysis of the produced biomedical data sets. It has become an indispensable part of the modern biological and biomedical sciences. Going forward, the advent of new experimental high-throughput methodologies, such as next-generation sequencing approaches, has led to significantly increased and highly complex digital biological datasets, thus requiring in-depth analysis to understand and interpret the information. Artificial intelligence and computer sciences have already been applied to modern biology and biomedicine, thus promising a new era in system biology that will enhance drug discovery strategies and facilitate clinical practices. The current lecture will define artificial intelligence and briefly describe the fundamental principles of the widely used machine learning, artificial neural networks, and deep learning approaches. Furthermore, we will investigate the role of artificial intelligence in major biomedical research fields such as genomics, proteomics, microbiome, chemoinformatics, and drug design techniques. Finally, this lecture will also highlight some challenges and future directions of artificial intelligence, ending with major concluding remarks. Section 2. What is artificial intelligence? Artificial intelligence refers to the ability of computers to to simulate the natural intelligence displayed by humans, to build intelligent systems that can function and run autonomously. Artificial intelligence includes a plethora of applications that can influence almost every facet of our lives, thus improving efficiencies and augmenting human capabilities. Major categories of artificial intelligence are natural language processing, machine learning, neural networks, deep learning, robotics, and computer vision. Artificial intelligence is divided into two distinct approaches: the generative whiteboard models that are interpretable as they encompass easy to understand algorithms. In contrary, the discriminative black box models describe algorithms with highly complicated mathematical functions, confusing for observers to understand. The technology of artificial intelligence harbors multiple methods, including machine learning and deep learning, which have already delivered many commercial products such as natural language understanding, machine translation, diagnostic systems, and search engines. Machine learning is a widely used artificial intelligence method that enables computer systems to understand and self-learn from information without being explicitly programmed. Machine learning algorithms build models based on a sample named training data to analyze the result, make predictions, and make decisions accordingly. Machine learning employs multiple approaches that can be divided into three major groups: supervised learning, unsupervised learning, reinforcement learning. Imitating the human brain, neural networks or artificial neural networks are composed of many interconnecting nodes and are aggregated into multiple layers. More precisely, neural networks can receive and recognize an input, and the multiple layer filter and process the information to provide a refined output. Deep learning algorithms combine machine learning and artificial neural networks and are based on building models that are trained by learning from processing labeled information. Although deep learning eliminates some of the data by pre-processing, which is typically related to machine learning, it consists of three or multiple layers, creating an additional type of artificial neural networks named deep neural networks. Additionally, computer science involves robotics, which aims to design machines that can substitute or assist humans. Computer vision uses deep learning models to train machines to capture and understand high-level information from the visual world, to perform accurate detection and classification of objects. This includes facial recognition, pattern detection, and image segmentation. Section 3. Artificial intelligence in biomedical sciences. Artificial intelligence has opened the door to entirely new opportunities in biomedical research, resulting in the development of new computational tools, with the biggest bets being to improve healthcare and medical diagnosis and treatment. A growing number of artificial intelligence applications that are available for artificial neural networks, such as genetic algorithms, deep learning, and other methods, aim to enhance genomic research, address drug development process, and support clinical trials. Advances in artificial intelligence are occurring across the spectrum of the biomedical fields, such as primary research, large-scale data analysis, target discovery, and personalized medicine. As far as the area of omics is concerned, the explosion of biological sequencing data has established machine learning as a key tool for many fields, including genomics, transcriptomics, proteomics, structural and systems biology. Numerous computational genomics tools have been developed to recognize a given DNA sequence, to predict genes as well as their regulatory elements, to determine their function, and estimate the location of protein encoding genes. BLAST is already counting 3 decades of life and still constitutes a simple and robust heuristic algorithm for the primary analysis of every DNA sequence, determining regions of similarity and mapping sequences to specific locations in the reference genome. In recent years, the study of protein secondary structures, folding patterns, functions, and interaction constitutes a challenge task for artificial intelligence. Both machine learning and artificial neural network methods are being used in the analysis of proteome and interactome. They can be categorized into two distinct groups: sequence-based methods and structural-based methods. In proteomics, machine learning software is used for the analysis of proteomic mass spectrometry data, for the detection and identification of individual proteins and the examination of their potential roles as biomarker for human diseases such as cancer. As for interactome studies, structure-based machine learning algorithms such as spot-on have already been developed for the prediction of hotspots residues contributing to protein-protein interactions in a human disease such as cancer. On the other hand, the evolving field of systems biology combines computational and mathematical analysis of complex biological datasets, aiming to identify molecular interactions and networks and understand tissue and even organism responses. Artificial-based algorithms can also focus on genomic and proteomic data analysis, with the main goal of predicting and treating diseases. Especially machine learning and deep learning approaches can identify and target the disease mechanisms to enhance therapeutic strategies. Bioinformatics analysis of the gut microbiome data from metagenomics sequencing experiments has led to the identification of many host diseases such as toxoplasmosis and irritable bowel syndrome. Predictive machine learning methods such as ANN algorithms have been established for the detection of microbiome host interactions connecting the gut microbiome with various human diseases. Artificial intelligence is very useful in the field of drug discovery, notably a large amount of biomedical data containing information ranging from primary DNA sequences, protein structure and function, to complicated interaction networks is necessary for generating novel drugs and new treatments. Cheminformatics databases such as PubChem, DrugBank, and Chimble are widely used public repositories that store a great amount of biomedical or biochemical and omics data that provide huge sources for artificial intelligence methods. Furthermore, multiple studies aim to combine information from publicly available databases to detect molecules that can represent promising candidates for the development of efficient drugs for various diseases. Over the past decade, machine learning and deep learning methods for molecule design have evolved and been developed to address target-ligand interactions and identify potential drugs. Machine learning methods manipulate the available data to design the molecular structure of the target and the potential drug. Machine learning is applied in all stages of the drug discovery process, starting from preliminary steps such as designing the chemical structure of the potential drug, investigating the drug's effectiveness, and finally the final FDA approval. Section 4. Challenges and future directions. Despite the widespread enthusiasm surrounding the incorporation of artificial intelligence in biomedical sciences, there are still numerous non-negligible challenges laying ahead that need to be addressed. To begin with, the precision and applicability of machine learning algorithms in different and complex real-life scenarios are highly dependent on the quantity and quality of the datasets used to train each model. Low-quality training data can reduce an algorithm's prediction performance. Furthermore, the black box problem represents one of the major concerns regarding deep learning approaches. More specifically, it refers to the inability to observe the decision process of an algorithm. Due to this non-transparency of how deep learning algorithms work, it becomes difficult to extract additional information, spot weaknesses, and make improvement of the models employed. Therefore, many efforts are being made to construct more explainable artificial intelligence models, as well as the development of methods attempting to interpret deep learning algorithms. Increased ethical concerns have arisen with the advent of artificial intelligence regarding matters such as the replacement of human workers, the protection of privacy, patient confidentiality, bias, and inequity. Section 5. Concluding remarks. Ultimately, the goal of artificial intelligence in biomedical sciences is a combination of data from different levels of omics information to uncover hidden aspects of cellular and molecular mechanisms of cells and tissues that are related to normal and pathological conditions. Under the prism of disease biology, artificial intelligence-based methods contribute to early detection and diagnosis, whereas machine learning and deep learning tools will encourage further drug design studies that will achieve both effectiveness and safety in future therapeutic strategies. It should be mentioned that artificial intelligent algorithms can perform tasks more efficiently than humans, can reduce human efforts, can reach a greater degree of precision, can work continuously, hence being available 24/7, can perform repetitive jobs, removing time-consuming tasks from humans. Even though bridging artificial intelligence and biomedical sciences show greater promises, the road ahead is still tortuous and full of substantial number of obstacles to overcome. Finally, we should be assured that artificial intelligence will augment but not replace human expertise. These are some references whose scientific information and figures have been relied upon in the preparation of this lecture. Thank you for your listening.